actually become memory cells. And these specialized white blood cells, when they transform themselves into the memory response, some of these memory responses are so powerful that they stay with us for years and years and years. Hopefully a lifetime, but for a long, long, long time. So definitely the creation of vaccines and getting booster shots and so on and so forth helps to kind of stoke number 4. Kind of helps create that memory in your body. When you go in and you get your booster, you're kind of reboosting that memory response. So yeah, we do come equipped with the memory response. So there's a lot of goals, but we know that a lot of this can go wrong too in so many different ways. What we're going to do though, so our immune system basically has 2 big branches. One's called innate and one's called adaptive. Innate is kind of very general. It's just kind of it covers kind of a lot of the general protective roles in our body. Adaptive is a lot more specialized. Use that word specialized. So we're going to talk about innate today, and I kind of like this graph because in a really simple way, it just kind of shows you the 2 big branches, innate and adaptive, how it's kind of split up. I stole it from another textbook though. So I don't think this is in your, I don't think there's an equivalent in this chapter. But on the innate side, what they did was they did subcategories. It says first line of defense and second line of defense. So that's what we're going to focus on today, is kind of that initial first barrier. And then we're going to focus on the white blood cells, which is the second line of defense. What we're going to do on Wednesday is talk about adaptive, and adaptive is a lot more specialized. Adaptive has other characteristics, and we're going to kind of compare and contrast the 2 in just a second. But here's where those special white blood cells that I said, especially that are tied into memory, fall underneath adaptive. On the innate though, it says first line of defense. So what are the first barriers? We have things like skin, right? Mucous membranes. What's kind of interesting is a lot of textbooks are including normal microbiota as a part of our immune system. Like just the fact that we carry around bacteria on our bodies and in our bodies, they're actually considering that as a protective role. So even just carrying those microbes that we do on a daily basis, that does help protect us a little bit against foreign invaders. Okay, so here's the compare and contrast that we're going to talk about. Innate and adaptive. And this is kind of a nice chart because it just only highlights the 3 most significant bullet points between the 2. So what we're saying is our immune system has 2 big branches, innate and adaptive. Do they work in isolation? Definitely not. Innate can talk to adaptive, adaptive can talk to innate. So they do work together. They're subdivided this way, but it's not like that they don't coordinate. They definitely coordinate together. So they work together. One of the big differences though is the response time. So how quickly they can act. Our innate branch, which we're going to talk about today, they're called the first responders. Why? Because they respond pretty quickly. So if there's some sort of foreign invader that enters your body, the innate is pretty good about swooping in fairly quickly and trying to contain, whether it's the virus, the bacteria, or the protozoan, whatever it is. Now adaptive will eventually respond too, but it takes time. That's why it says second to respond. So it does take time. In fact, I kind of gave you guys a little bit of a hint about that, because remember we talked about HIV, and I said that usually on average it takes about a month for you to start making antibodies against HIV. So if you go in and get your HIV test, you'll usually, give a false negative if you've been exposed. That's what I mean when it says second to respond. So our adaptive, it does material. And I'll give you examples when we talk about this. The only thing that's kind of difficult to remember is the name of these little receptors that they use to recognize the foreign material. It has a really weird name. So they're called toll-like receptors. And the reason why they call them toll-like receptors is because they discovered them in a fruit fly, so they have this really strange name. On the adaptive side, now the adaptive is so special. Okay, so the adaptive is so specific. The reason why it takes so long to respond is because it literally learns every tiny, itty-bitty detail what you're infected with. So, like for example, HIV, since we just talked about it, it's kind of a good example to use. On the adaptive side, your adaptive immunity will literally figure out, okay, GP120, I'm going to attack that. GP41, okay, that's encapsulated. It literally finds every single little detail, and it mounts a response against that tiny little molecule. So it's really specific. And then on the bottom is the memory. Our innate cannot remember slot. So when we talk about a lot of the white blood cells today, like we're going to talk about macrophages, they can't remember anything. All they do is just attack. So if you're sick with something, they'll attack it. Will they remember it in the future if you got sick with that same thing again? No. They're totally useless. But on the adaptive side, luckily, those very special subset of white blood cells, we're going to talk about the special ones, those ones can remember. So as long as those ones get involved in the reaction, then you'll get the memory covered on that part. Now the bummer is, we're going to talk about this later on this semester, the big bummer is things like salmonella, not to be Debbie Downer, but it's kind of Debbie Downer here. Salmonella, there's so many varieties of salmonella out there. So if I were to get sick with salmonella today, my body would remember it. But since there's so many varieties, if I get sick with the other variety next week, I just get sick all over again. Like the memory only protects me against that one specific type of salmonella. It doesn't cover all of the salmonellas out there. So I just get sick again, and then again. So the memory is great if there's not a lot of varieties of that microbe. But if there's a lot of variety, it's kind of hard because you just end up getting sick. So we're going to talk about the first and second lines of defense. Just briefly, we'll mention the first line. What we're going to do is focus on the second line. All right, so first line, what are those initial physical barriers that we come, quick with to protect us against a variety of microbes? Definitely skin is so important. Skin is in there. So under first line of defense, skin is one of the most important physical barriers that we have. One of the organisms that we're going to talk about later on in the semester, burn patients, they're really susceptible and vulnerable to picking up a variety of bacterial infections. So intact skin, we have proteins like keratin, we have epithelial cells, all of which I'm sure you guys learned about in anatomy, right? They're all tightly packed in there. Usually there's protein junctions that help keep everything tightly bound together. That's all helpful. Sebum, does anybody know what sebum is? Or what is sebum? Yeah, I heard somebody say it. Oil? Yeah, oil. But yes, physical barriers are really, really important. We probably take that for granted. Besides those two things, we also have flushing mechanisms. So we have fluids, we have ways of flushing out potentially foreign things that have entered our bodies. Tears, you know, the production of tears, that helps to sweep out foreign material from protecting our eyes. Saliva, since that's a portal of entry into our GI tract. It's not perfect, but we do have some enzymes in our saliva that can potentially digest foreign material. We're going to talk about the cilia in just a second, but even our sweat comes equipped with enzymes. Lysozyme is an enzyme that we're going to mention also. The flushing mechanisms of our urine, right? So, flushing out the genitourinary tract. Jalen kind of mentioned that with healthcare professionals getting urinary tract infections because they're always holding their urine at work. So yeah, urination, defecation, even the vomiting reflex for flushing. But we're going to highlight some of the enzymes and then also just mention the cilia really briefly. Since we're moving into flu season, also at the end of the semester we're going to talk about the flu virus and how the flu virus works so quickly at entering your respiratory tract, and it has such a damaging effect on your cilia. So I have a picture of our beautiful packed cilia layer, and it lines our airways, lines our respiratory tract, and all of these cilia have a beautiful sweeping motion, and definitely there's different things that can damage that sweeping motion of our cilia. So one of which we're going to talk about later on is the flu virus. So if you get sick with the flu, the virus actually works really quickly, and it goes in and actually infects these epithelial cells lining your airway. One of the things that it quickly does is it starts killing off all of your cilia. So all these cilia actually just get annihilated, and you lose that sweeping mechanism. Now for all of us that are kind of, younger, healthy individuals, you feel really bad, you get sick with the flu, and then eventually you recover, right? But if you're older or maybe really, really, really young, the problem is once you lose the cilia, which we're saying is a part of your immune system, without that protective sweeping mechanism, it makes you susceptible to picking up something else. Maybe another bacterial infection on top of your flu. And the problem is those organisms that are linked to pneumonia, so like Streptococcus pneumoniae, right, or Klebsiella pneumoniae, these pneumonia-causing bacteria. Now you're susceptible to these other bacteria. They enter. People can develop pneumonia on top of their flu. Usually that's the deadly, the deadly part of getting the flu, right? All right, and then we're going to mention some of the enzymes. Just one in particular. Mention the gastric juices. Oh, did they fix the clock? Wow, this is like amazing. I asked somebody to fix it and it got fixed. Pretty cool. I was like, what? Okay. Gastric juices and lysozyme. Okay. So unfortunately, we do know we come equipped with a stomach full of gastric juice, but there are a lot of microbes, unfortunately, that can still sneak through and survive and pass through the stomach and then make their way into our small and large intestines and still make us sick, right? But it is an important barrier. Usually we're talking about a low pH, so it's very acidic. Definitely that helps. It's not perfect, but the acidity is very helpful. So one of the things we talked about in lab is we don't know of any bacteria that can just live in our stomach because it's so acidic, except that Helicobacter pylori. And we also know there's digestive enzymes, a lot of which I'm sure you memorize in anatomy, maybe, or physio. So digestion starts in the mouth, but eventually in the stomach you get bombarded. Now these digestive enzymes are mostly for food, but they can have an impact on microbes that are entering. And then we do know there's molecules that get secreted by the stomach. In very general terms, they're called defenses. I don't want to get into like a lot of examples, but there are molecules, other molecules besides just enzymes, that also, what they do is they target microbes as well. So there are other molecules that get dumped out, and they're looking for foreign material to attack. So all these things with enzymes, acidity, molecules getting dumped out. We even have in our body, though, an enzyme called lysozyme. And lysozyme is an enzyme. We technically mentioned lysozyme when we talked about the phage, those bacteriophage. Remember we said they attach, and then they kind of squat down and they dump out lysozyme? And lysozyme is what drills a hole through the peptidoglycan. So this is actually an enzyme that breaks down peptidoglycan, digests peptidoglycan. So that's another nice enzyme, and that enzyme is actually made in our bodies by a lot of our bodily secretions. Saliva, we have it. Tears. And what's even cool is our sweat. Even comes equipped with lysozyme. So that's another helpful enzyme that can attack peptidoglycan. Now, unfortunately, lysozyme is probably not going to do so well against gram-negatives, because gram-negatives have that outer membrane, but at least for gram-positives, right? Because gram-positives, they have their peptidoglycan very exposed, so they'll be really susceptible to lysozyme. All right, so what we're going to do is focus on the white blood cells. White blood cells. I have here types of cells in our blood. Definitely, as far as the most numerous type of cell we have in our blood, it's the erythrocytes, the red blood cells, right? And we kind of mentioned them when we talked about malaria. So going back to the malaria section. What do the red blood cells do for us? What's like their number one job? Oxygen. Yeah, they pick up oxygen, right? So they grab oxygen and they transport it around and dump it off to our tissues, which is so important. So that's why malaria is so devastating, because our white blood cells are getting attacked by Plasmodium. We're losing those red blood cells. People can become very anemic, which is bad. But yeah, we have platelets. Platelets are also important, kind of in that clotting pathway. But what we're going to focus on are the white blood cells. Now, that white blood cell can be used interchangeably with the general term leukocyte. So leukocyte is a very general term. And we have it subdivided into what's called granulocytes and agranulocytes. The reason why there's those two subcategories is it just has to do with the way they look. So I'll show you pictures. We have a lot of white blood cells that are called granulocytes because they look really grainy underneath the microscope. That's all that means. So it has to do with more of an appearance, how they appear underneath the microscope. Agranulocytes, they have a little bit more of a smoother appearance underneath the microscope. Okay, so I have a picture of some examples of the white blood cells we're going to focus on. Now in my picture though, in my picture here, remember. They come equipped with that memory response. Okay, so what I have is kind of the main white blood cells that we're going to cover. We're going to make a chart on the board, but these are kind of the big categories at least. We're going to talk about neutrophils, basophils, eosinophils, mentioned monocytes, and what it means to be a macrophage. The only thing that we're not going to get into in a great detail are these NK cells. They're a type of killer cell, natural killer cell. We're mostly going to focus on the rest. But these are all under our innate immune system. Not the first line of defense, but the second line of defense under innate. So if a pathogen were to slip through your skin or mucous membrane, we would have these blood-borne cells that would kick in and respond right away. Now, definitely one of the things we're going to emphasize is phagocytosis. Phagocytosis is probably one of the important jobs of the innate immune response. They are looking to eat, cellular eating. So they're looking for foreign material to eat. And this is something that generally that adaptive response does not do. They don't really perform the phagocytosis as well as the innate. It's really the innate that does the cellular eating part. Now, inflammation, as painful as that is for us, because we get redness and swelling and heat, and it becomes very uncomfortable and very painful. But when inflammation kicks in, that is your body's response. It's trying to mount a response and attack the foreign material. So when our tissues get inflamed, that means your immune system is kicked into high gear. It's actually responding. And then fever technically is in here also. So when we develop a little bit of a fever, again, that's your innate immune response working hard and responding against the foreign pathogen. And it's trying to tell your body to increase its body temperature to try to ward off that foreign material. And then we're going to mention complement. Cytokines we're not going to get into now. We'll talk about it more on Wednesday. But remember, cytokines, we said, are those chemical messengers. So our immune system, they talk to each other all the time. They send messages to each other. They use chemicals. One example we gave is that TNF-alpha. That was one example of a messenger that gets dumped out by your T cells. All right, so let's make our chart. Go. Keep this up. And you guys, do you cover white blood cells in anatomy? That's good. So hopefully it'll be a little bit. Some of it will be a little bit redundant. All right. So we'll make our. One class, 16 weeks, just talking about this, because there's that much material, and then and adaptive. I was like, a little bit boring. But we're going to make our own chart. We're going to highlight kind of the big bullet points. We're going to do the mention. Let's see. I'll do a granulocyte. Do that first. There we go. And then the percentage. Specific type will be type of white blood cell. Then do the percentage. And then the function. Do you guys get to look at white blood cells in anatomy under the microscope?
Yes.
Okay, so you look at them a little bit. So let's start off with the neutrophils. All right, so neutrophils are in there. What's a little bit confusing is, I would say really most textbooks call them neutrophils. Sometimes though you see in other textbooks, they'll use this term PMNs. And PMNs and neutrophils, you can use those terms interchangeably. They're talking about the same cell. So PMN stands for polymorphonuclear cells. So it's got PMN. Polymorphonuclear. The reason why neutrophils are sometimes called polymorphonuclear is because they have a nucleus, but the nucleus is kind of a weird shape to it. So under the microscope in anatomy, neutrophils are really easy to find because their nucleus, there's only one nucleus, but it kind of has this polymorpho shape. The shape is all kind of what we call lobed. So the nucleus kind of has like these multiple lobes that kind of bulge out, and it makes it have a very distinct appearance. So that's why the nucleus, we say, is polymorpho. It's kind of morphed into these different lobes. All right, as far as percentage though, neutrophils have the highest percentage out of all the white blood cells. On average, it can be as high as 70%. That is the highest. So on the slides in anatomy, if you're looking for a neutrophil, you're going to find plenty of them. They're all over the place. They're really easy to find. Okay. The neutrophils, they are the first responders. They respond really quickly. Usually they're the ones that arrive to the site of infection the fastest. So what we say is that they're really very mobile. They can move around very quickly in your blood. Not only that, but they don't just stay in your blood, they can escape from your blood, can leave blood and enter your tissues and attack any foreign material that's hanging out in your tissues. On the downside, they don't live very long, so we call them very short-lived cells. What that means is they'll go ahead and get there and they'll start attacking, but eventually they'll die pretty quickly. As they start to die, usually these are the cells that you find in pus, right? So they're found in pus. So pus is usually kind of a collection of dead white blood cells, and neutrophils are usually the main one. All right, as far as their job, the most important job is phagocytosis. They're really, really good at phagocytosis, at cellular eating. They're probably one of the most effective at phagocytosis. They do it really well, so that's good. So they will actually cruise around and eat the foreign material. All right, so they're the first responders. They're nice to have. We do know that people that have like genetic disorders that don't make enough neutrophils, or they don't work as they should, they get really, really sick. So the neutrophils are really important. All right, the other granule site are the basophils. So we'll go to the other extreme, basophils. So we'll go from the highest percentage to the lowest percentage. Basophils can be as low as like 1 to 2%, really low. This is the lowest. So if you're hunting for a basophil in anatomy, it takes you forever. A lot Allergies, yes. This is linked to allergies. We're going to talk about allergies later on. So yeah, histamine is what triggers that allergic response. Now histamine is nice to have around because histamine triggers inflammation, which is good. It's a good thing. It activates inflammation. Some inflammation is good because it means your immune system is working hard. It's an overdrive. The problem is we're going to talk about allergies later on in the semester. Allergies sometimes are those allergic reactions are so dramatic that they can actually cause a lot of harm, which is bad. All right, and then the other thing we'll mention about the basophils is they cannot perform phagocytosis. So they're really good at kind of cruising around and dumping out chemicals to help ward off infectious agents, but their job really is not to phagocytose, not to eat anything. So they'll help out by releasing their chemicals, but that's pretty much it. Okay. All right, so we'll mention the last granulocyte. My last one is the eosinophils. They're also in that granulocyte category. Eosinophils are also easy to spot in the microscope. Their nucleus is not as polymorpho as the neutrophils, but we always say that eosinophils look like aliens because the nucleus kind of has like this, like alienized type appearance. So it almost looks like a really distinctive boomerang a little bit. All right, and eosinophils, as far as their percentage, they're pretty low also. Not as low as the basophils, but they're pretty low. So we're saying like around 4% is usually the typical eosinophil count. Okay, as far as their functions, they are also phagocytic. So they can also perform that cellular eating, not as well as the neutrophils. Neutrophils are kind of predominantly, but they can eat. They also store chemicals in their little granules, just like the basophils. And where the eosinophils come in pretty handy is they are most effective against helminths, the worms. So we'll mention this later on in the semester. So if you get infected with a helminth, right, those worms, the eosinophils are primarily the ones that swoop in and they start responding. And they'll start dumping out their chemicals to try to attack the worm. So definitely any sort of like parasitic infection, the eosinophils are handy for those. All right. So those are the only 3 granulocytes that we're going to mention. And then we only have one big category left. So the last category are the A granulocytes. A granulocytes. And we're going to mention the monocytes. The monocytes. All right. So it's the same cell, it's just one is inactive state and the other one's the activated state. We call it a macrophage. And the percentage of macrophages is actually pretty high. So these you can generally find without any problems. Generally it's around like 8% or 10%, so it's close to 10%. So generally you don't have too many problems finding a monocyte or a macrophage. What's really nice about the monocytes / macrophages is the monocytes, we have them permanently in different fixtures in our bodies. So there are, quote, wandering monocytes, where you find them in your blood. And all they do is wander. They just travel through your blood looking for foreign material. And we also have what's called fixed monocytes, which are really nice. So we have the wandering ones, the fixed ones, they just stay in your tissues. So it's nice, you kind of have both. Some that float around through the circulation, some in the tissues. We have some permanently in your lungs, permanently in your liver, GI tract, all sorts of different areas. But we're going to talk about this with tuberculosis with the lungs, because when you get exposed to the tuberculosis bacteria, it's the fixed ones in your lungs that try to attack the tuberculosis bacteria. All right, their primary job, really, just like the neutrophils, is phagocytosis. And just like the neutrophils, they do it very well. So once they're activated into macrophages, these macrophages will go ahead and they'll eat cellular eating. There is a special function here, though. Neutrophils, what they do is they just eat and then they die, because we said they're so short-lived. What's nice about the monocytes, though, is these are a little bit longer living, so they don't die right away, which is really nice. And what they do is they live long enough after they eat the material. Just add a couple quick things up here. After they eat their material, they'll take that foreign material and they'll actually deliver it to your adaptive immune response. That's kind of the other nice thing about the macrophages. Just wanted to add a couple quick things. All right. So for the macrophages, not only are they longer living, but they actually can present the foreign material to your adaptive immune response, to the adaptive branch. That's actually a really powerful function, super powerful. So we're going to talk about that in just a second. Innate is mostly working with phagocytosis and those chemicals in the granules to try to contain the infection. Monocytes, though, are super important because they're the main ones that will actually communicate with the adaptive and tell them, here's the material that you're sick with. I'm going to drag you into the response and get them involved. So then now, finally, hopefully we get our memory part that kicks in. So they're the presenters. We call them presenters, antigen presenters. And then lastly, the last thing. Let's see. Lastly is that these are lymphocyte. But technically, lymphocytes are in the adaptive branch, so they're not in the innate side, but we're just going to briefly mention them here. All right, so we're going to mention the one type that are called T helper cells. T helper cells. Now, we call them T helper cells, but what most people call them is they call them /CD4 T cells. The reason why is because if you are that type of T cell, you have a very distinctive protein on your surface called CD4. That's just a protein called CD4. And this is one example of a lymphocyte under the adaptive branch. What the T helpers do is all they do, I call them wedding coordinators because that's literally what they are. They don't kill anything, so they cannot kill. They don't have a killing effect. They also definitely cannot phagocytose. They can't eat anything. But what they do is they tell everybody else what to do. So they're called the helpers. And the way that they function is they usually release cytokines, chemical messages that coordinate other white blood cells. So they don't kill anything themselves, but they're really, really important because they coordinate the rest of your immune response. We definitely need them. And this was the white blood cell that we said HIV really primarily has a high affinity for. It really likes attaching to that CD4 protein, right? So that kind of kickstarts the infection. All right, and then the other a type of T cell, we call them the T killer cells. And these actually do kill. They're not called CD4, they're called CD8 positive T cells. So the killers, they have a different protein called CD8 on their surface. And finally, so here's where we get our killing effect. The killer T cells actually kill. So they can kill infected cells. If you have an infection going on, if there's something hiding out in your body, the killers can actually swoop in and go ahead. We're going to talk about, they're going to dump out chemicals and will actually kill the infected cell. And they also come in pretty handy. They are helpful against tumor cells, so cancerous cells, because they have that killing effect. So that's good. All right, and then I just have one more left, and then I think we're done. So we have the helpers, the killers, and lastly, my category of lymphocytes. There is another last type of T cell. We call it a specialized helper T cell. So it's a type of helper T cell, but it's different. It's like extra special. The reason why is it gets a different name. They're called regulatory T cells. So they also have that CD4 protein on them, but they're extra special. The reason why these are so extra special is because this is a T cell that actually tells the rest of your immune system to stop. So these T cells actually coordinate the stoppage of your immune response. They tell your immune response to stop. damage. So they will actually protect you against damage. Protects body against damage. And when I say damage, we know damage can come in the form of, for example, I'll just say autoimmune disorders. Right? We want our immune system to react, but if it acts too powerfully, it can actually start hurting your own body. It can kind of turn against you. So it's a little bit of a delicate balance. So the helpers kind of help with that. They help protect you so that your body actually doesn't start getting damage to itself. So that's where the stopping is good. So, all right, I think we've summarized just about everything. We talked about granulocytes, agranulocytes. Oh, sorry, I forgot. I was like, wait a minute. There's one last one that I'm going to mention under lymphocyte. I totally forgot. And you probably mentioned them in anatomy, so we don't just have T cells. We have one more lymphocyte called a B cell. B cell. So it's not just T cells. So under lymphocyte, the 20 to 25% can include the helpers, the killers, the regulatory, and then my last one, it also includes the B cells. Forgot the B cells. B cells. So that 20-25% includes all of those lymphocytes. B cells, all right. So it's not just T cells. We also have B cells. B cells, I don't know if you guys remember, but they make antibodies. They make antibodies for us. We'll talk about antibodies later on. But antibodies are super powerful. So B cells, these, all right. Talk about antibodies. We're going to talk about them because they're also under adaptive. Okay, so we'll have more time to focus on those, but I forgot to mention that the B cells are in there. All right, so now that we have the white blood cells kind of covered, my last couple things. Alright, we said basophils are important for allergic reactions. We're going to talk about that later on. Neutrophils are the first responders. Eosinophils are for worm infections, help protecting you against worm infections. The other thing that I didn't mention is how do all of those white blood cells on the board that we just laid out, how do they actually recognize? Because remember we said there's a survey function, there's a recognition function, then there's the attack function, then there's the memory function. So the recognition step is so important. And basically, on the white blood cells that we charted that were under the innate, so the neutrophils, the macrophages, basophils, they all come equipped with these little antennas, these receptors, that allow them to recognize the foreign material. What's pretty cool is we have kind of these generic receptors already. They're already on our white blood cells. They're ready to go, and they're just waiting for foreign material. And each one recognizes a different piece of foreign material. So you guys actually already know a lot of these. Like this receptor recognizes flagellin, and this receptor recognizes LPS. And this receptor is looking for teichoic acid. So all these things that we talked about, flagellin, LPS, teichoic acid, those are the materials that the receptors are just waiting for. So once any of that material enters your body, they respond really quickly. They use these special receptors. They have a weird name though because they're called toll-like receptors. And they discovered these receptors originally in a fruit fly, and then they figured out, oh wait, humans have them too, so that name stuck. And all these receptors are on every single white blood cell that we have in our body. Not the T cells and the B cells, but everything else. So once these make contact with that foreign material, it'll activate that white blood cell and it'll start either phagocytosing or dumping out chemicals, all to attack the virus. I think one of them down here, like these down here recognize viral RNA such as HIV. So we have a lot of different these receptors. So once the contact happens, like for example, they're showing a little bacteria, and the LPS receptor says, oh, there's LPS, then it stimulates this white blood cell, it responds and starts dumping out chemical messages, and that's what kind of gets your immune system going. So here's those cytokines, right, that we kind of mentioned a little bit, just briefly. TNF alpha was the main one that we mentioned, but there's other messages that get dumped out. And then we already mentioned regulatory T cells. We already mentioned this on the board. We said this was the specialized T cell, and it was a Japanese scientist who became super famous for discovering that specialized T cell, because we didn't know about that specialized T cell for a really long time. We didn't know it existed. And then we figured out that, oh wait, not every single white blood cell in our body is there to just attack or respond or be involved in inflammation. That there is a type of white blood cell out there whose job it is to stop, to kind of get your immune system under control eventually, and to inhibit the activity of other white blood cells. So that was kind of a big deal, because we always thought every type of white blood cell was meant to just attack or phagocytose or do something, not to stop the immune system. So this is kind of like a big cell that's the focus of a lot of research. A lot of cancer research labs are interested in these specialized T cells, and then also other labs that are tied to autoimmunity, because we know that for people who have autoimmune disorders, auto means what again? Self, yeah. So your immune system starts attacking your own body, right? So for autoimmune kind of research people, they're interested in these regulatory T cells because they're trying to figure out how can we make them better for people who have autoimmune disorders. Their immune system's out of whack and they're actually attacking their own bodies. So how can we make this function even better for those individuals? All right. In my lab, we were working on the other side. We were working on the cancer side. So we were trying to figure out ways of trying to come up with ways of actually like stunning the regulatory T cells. So we didn't want to enhance them. We actually wanted to block their function. So I told you guys their function is to stop, right? They're trying to tell the rest of your immune system to stop. And what we wanted to do is we wanted to temporarily stop the function of the regulatory T cells. So by kind of like removing the stop sign, we wanted to unleash your immune system and get it really kicked in the high gear so it would attack cancer cells, right? So it's a little bit weird because this is a good type of T cell to have around, but we wanted to temporarily remove them from the picture so that your immune system would kind of be unleashed to attack cancer cells, right? So I have a picture of what I mean by that. So like my project directly, we worked with mice, which was, I like animals too, but we had groups of mice, and I'll show you a picture of kind of like the experiment that I set up. But we had groups of mice, and remember in the virus lecture we said there's that cell culture. You can grow cells kind of like in a plastic petri dish. So that's what we did. The type of cell that we grew was the type of colon cancer inject these colon cancer cells on top of their backs so that we could see the tumors like actually growing, right? We'd wait a little bit of time, and then we would try different treatments to see which ones would work and which ones were not so great, just to get an idea. But the type of treatments that we came up with, again, we were trying to stop the regulatory T cells, to kind of temporarily get them out of the way, so that there wouldn't be anything there to inhibit your white blood cells. We wanted to unleash your white blood cells so they'd attack like crazy. And this is actually my picture of my groups of mice. They're actually alive. They're just sleeping. The picture, I promise. They are alive. But we made them really sleepy so they stay still for the picture. So this is the way this works. Unfortunately, in this group, this was my control group. So in the control group, what we did was we treated them, but we treated them with an antibody that we knew was not a very effective antibody as our negative control. So you can see on their backs, the colon cancer cells grew like crazy, right? The tumors are huge. And then you'd actually measure them with a ruler. These other two groups, though, what we did is these are the treatments that temporarily stopped the regulatory T cells. So they temporarily kind of got those out of the way. And then when we kind of unleashed the immune system, there wasn't anything around to inhibit them anymore, so they just responded like crazy. And they started attacking the cancer cells like crazy, to the point where a lot of these mice, some of them became tumor-free, like their tumors were actually like totally gone, or they shrunk a lot, like one or the other, right? So that's kind of like what we did with the different treatments. So that's kind of like one good application of how that works with the regulatory T cells.
So what exactly did you use the treatment with that classified?
Oh, no, that's a good question. These are basically antibodies. Yeah. We tried to engineer antibodies that would like attack those regulatory T cells and kind of temporarily get them out of the way. So that's what they are. They're antibodies. We treated them with antibodies.
Is it like subcutaneous or is it like injection?
Oh, yeah, so that's a good question. These are all given intravenously. Okay. Yeah, so that's the other difficult thing. You're like trying to find a vein on a mouse, trying to hunt that vein down, and then you're injecting the antibodies like right into their veins. Yeah. So generally, you kind of like heat them up under a lamp to get nice and warm. And those veins kind of pop out. And then try to find that one, and try to get that antibody in here. It is hard though, yeah, finding a vein on a mouse. That's a good question. I forgot about those days. Alright, so my last couple things I'll mention for today, and that'll be it, is phagocytosis. Phagocytosis, you guys already know, is cellular eating. So cellular eating is so important with innate immunity, so important, because especially not every white blood cell can phagocytose. Neutrophils and macrophages are like notorious for doing it. Eosinophils, we said, they can do it too, but really it's the neutrophils and the macrophages. And the cellular eating is so important because this is the step that is really like the first response that's carried out by our immune reaction is phagocytosis. So it really starts with phagocytosis, and then everything else will respond accordingly. But you kind of really need that initial phagocytosis to kind of get the rest of your immune system responding. So, and we're going to go over just quickly the steps involved in phagocytosis. But definitely, so this is all about cellular eating. Actually, it's all tied into the first PowerPoint. There's a survey function, a recognition function, like a destruction function. For phagocytosis, though, these cells, there's no memory, though. So neutrophils great at eating. All they do is eat, but they never remember what they ate. So there's no memory, unfortunately, tied in with our neutrophils and macrophages. They're just looking for things to eat. But we did say that the eating is facilitated by those toll receptors. So they're using their toll receptors to find flagellin, LPS, capsid, whatever it is. And this is probably the best picture for showing you all the steps involved in phagocytosis. Remember in chapter 4 I mentioned we were going to come back to lysosomes. Lysosomes, that organelle, because remember, what did we say the lysosomes, what do they do? They digest, right? Yeah, so the reason why we kind of pointed that out is because it's tied into the phagocytosis process here. So our innate immune system cells, the ones that we highlighted on the board, they're scavenging around, or they're fixed in our tissues, and once they meet up with foreign material, their membranes, they form like a little pouch, and they literally grab the whole bacteria or the whole virus or whatever it is, and they literally eat the whole thing. So the whole entire material is ingested. We say ingested. It's taken in by the white blood cell, and it's kind of sequestered in a little pouch. What's a little bit funny is they give a name to that pouch. That pouch is called a phagosome, and then that pouch, what that pouch ends up doing is that pouch will eventually meet up with the lysosome, that organelle. The two actually meet together, and when the two meet together, we call that a phagolysosome. But the phagolysosome is now, that's literally the step where that material is digested. It's digested. We get the digestion part. So the enzymes stored up in the lysosome will work to attack and digest. Now what's pretty cool though, actually, it doesn't say in this picture, those lysosomes, not only do they just have digestive enzymes, but they actually have those free radicals in them too. Remember those free radicals we said were so bad, like hydrogen peroxide and that superoxide free radical? Those are actually stored up in the lysosome also. So free radicals also attack the bacteria like crazy. Even though it can make a little bit of catalase or whatever it is, it gets bombarded by all these free radicals, and that helps to kill off the microbe. Eventually it comes out and the white blood cell just spits it out. It spits it out at the end. But what it doesn't show in this picture, I'm going to add it to the very, very end. In the picture, it just has the white blood cells spewing it out here at the end. After it's digested, all the little remnants, step number 7, it says discharge of waste. So the waste kind of just gets released out. But one thing I wanted to add to this though, because we're going to talk about this, remember on the board we said macrophages though, they'll eat the material and they'll digest it and they'll release the waste, but macrophages can do one extra step beyond this, and that one extra step is they macrophages. Because it's not neutrophils, it's macrophages that do this. Macrophages, not only do they do all the steps and they discharge the waste, but they also can present the material, to your adaptive side. Adaptive immunity. So that's kind of the cool part is, even though this is all innate, the macrophages, they don't just dump out the waste and then they're done. They'll actually grab a little bit of this waste and they'll actually start displaying it. When I say display, they'll show it. They'll actually intentionally show off this foreign material, and they'll coat themselves digested, or just avoid your white blood cell altogether. So it can prevent the white blood cell from attaching to it, or it can, instead of staying in that pouch like it should, I said there was that phagosome pouch that it should be sequestered in, sometimes microbes just slip on out, and they totally just avoid being sent to the lysosome organelle. Or they do actually make it to the lysosome, but they prevent the two from fusing together. So step number 2 and 3, they never get digested. Or the microbe just full on just kills your white blood cell. It just finds a way to lyse it and kill it. So it kind of overpowers your white blood cell. So unfortunately, we do know there's a lot of microbes out there. They find a sneaky way of just avoiding the phagocytosis altogether, which is not good. Because then that means we're probably gonna get sick. So I'm gonna stop there, guys.